Hi, and welcome to 6.2.2 Synaptic Transmission. This is the spec if you want to stop and have a quick read of it. So, a synapse is a junction between neurons which takes a signal in one direction. We say that anything going in one direction is unidirectional. At the synapses, neurotransmitters are used, and the neurotransmitters help to transmit the signal from one neuron to the next. And examples of neurotransmitters include acetylcholine and noradrenaline. You don't necessarily need to know specifically lots of neurotransmitters, but they sometimes come up in exam questions. You just need to be able to apply the principles of them. Okay, so this diagram here shows all of the different parts of your synapse. So you've got your presynaptic neuron, and then this is your postsynaptic neuron. Now we call this bit of the presynaptic neuron the synaptic bulb, and then we've got a presynaptic membrane here. We've got a postsynaptic membrane. We've got the synaptic cleft here, which is the actual gap in between the two neurons. And then we've got these vesicles, and within these vesicles, you've got your neurotransmitter. So let's take a look at what's happening. So at number 1 on your diagram, let's get a pointer. Number 1 on your diagram, the action potential is coming from the axon and reaching the synaptic bulb. So that action potential is coming down and reaching that synaptic bulb. When the action potential reaches the synaptic bulb, it causes calcium channels within the membrane of the synaptic bulb to open. And then calcium is going to flood in to the synaptic bulb. What that calcium does is it then causes these vesicles containing the neurotransmitter to move towards the presynaptic membrane. Here you can see that these vesicles have reached the presynaptic membrane. You don't need to know about this docking protein for A-level, so don't worry about that. And at step 6 here, the neurotransmitter within the vesicle is being deposited into the synaptic cleft by exocytosis. So all of that neurotransmitter moves into the synaptic cleft. Then this is a mark that people always forget in exam questions. So the neurotransmitter diffuses across the synaptic cleft. So exocytosis diffuses across, and then once it gets across to the postsynaptic membrane, it's going to attach to the receptors on the postsynaptic membrane, and this causes the sodium channels to open. Now, once the sodium channels open, sodium can flood into your second neuron, and that then causes a new wave of depolarization or an action potential as long as the threshold is reached. So you want lots of the sodium channels to open. You want lots of neurotransmitter to bind to lots of receptors, causing lots of sodium channels to open, and then enough sodium moving in order for depolarization to occur. And then once all of that has happened, the neurotransmitter that's left within the synaptic cleft or with attached to the receptors leaves the receptors, and within this area there's enzymes that can break down the neurotransmitter. So they break the neurotransmitter down, and then the product of that breakdown are reabsorbed back into the synaptic bulb, and then they're reformed back into neurotransmitter and used again. And the reason we need to break the neurotransmitter down is so that you don't get a continuous, the action potentials aren't continuously fired. So once the neurotransmitters have done their job, they're broken. Okay, so there's different types of neurotransmitter. You can either have excitatory neurotransmitters, and they cause the depolarization of the postsynaptic membrane. Therefore, an action potential can be fired. Or you can have inhibitory neurotransmitters. They cause hyperpolarization, and if you remember from nerve impulses, any membrane that's hyperpolarized can't be depolarized. So if they cause hyperpolarization, it means that depolarization can't occur, and therefore no action potentials can be fired. Adrenaline is an example of an excitatory neurotransmitter, and there's one called GABA, which is an example of an inhibitory neurotransmitter. You don't need to know them. There's GABA is in the stem of some, and adrenaline are in the stem of some of the questions, but you don't need to know about them specifically. And some neurotransmitters can be excitatory or inhibitory depending on the type of synapse and where the synapse is. So, for example, cholinergic synapses use a neurotransmitter called acetylcholine, and in the central nervous system, this is acetylcholine is excitatory, whereas in the heart, it's inhibitory. So some of them can be both depending on the location. Okay, summation is the effect of all the neurotransmitter that's released into the synapse added together. So sometimes you might have, you can see here, this is a dendrite, so this is where this would be your postsynaptic neuron, and these ones here are all of your presynaptic neurons. In spatial summation, it means that you've got more than one neuron that is synapsing with the postsynaptic neuron, and it means that here we're going to have 3 times the amount of neurotransmitter. So the neurotransmitter is released at this synapse, and this synapse. And what that then does is it increases the chance of more sodium channels opening, and therefore the chances of reaching the threshold for an action potential to be fired also increases. So the more neurotransmitter that's released, the increased chance of reaching the threshold. Now, temporal summation, you can see here, what you've got is one presynaptic neuron and your postsynaptic neuron here. What's happening is you're getting lots of action potentials being fired in quick succession. So that means that every time that action potential is fired, some more neurotransmitter is deposited into the synaptic cleft. So therefore, we're increasing the amount of neurotransmitter in the synaptic cleft. Therefore, we're increasing the amount of sodium channels that are going to open in the postsynaptic membrane, which then increases the chance of the threshold being reached and therefore an action potential being fired. So spatial is 2 or more neurons that are synapsing with this postsynaptic neuron, and then temporal is an increase in frequency of action potentials along one presynaptic neuron. Okay, the next bit is all about how different drugs affect synapses. So this comes up in exam questions quite often because it's quite a nice application. You don't need to know specific drugs, but you need to know how different types of drugs can affect the synapses. So agonists have a similar shape to the neurotransmitter. And because of this, it means that they can bind to the receptors on the postsynaptic membrane, and they therefore mimic the effect of the neurotransmitter. So if you take an agonist, it will do the same job as a neurotransmitter, attach to the receptor, cause the sodium channels to open, and therefore the sodium moves in, increasing the likelihood of an action potential being fired. An antagonist binds to the receptors, but it blocks the neurotransmitter from being able to bind. So it has a similar shape to a complementary shape to the receptor. It will bind to the receptor, and then it will stop the neurotransmitter binding. Therefore, the sodium channels aren't going to open, so sodium isn't going to move in. Therefore, the threshold isn't going to be reached. Action potentials can't be fired. Enzyme inhibitors, they inhibit the enzymes that break down the neurotransmitters. So those enzymes that are within the synaptic cleft that hang around there waiting to break down neurotransmitters are inhibited by these drugs. This means that the neurotransmitters can stay in the synaptic cleft, continue to bind with the receptors, and therefore action potentials will continue to be fired. Some drugs can stimulate the neurotransmitter, so it just causes more neurotransmitter to be released. Obviously, the more neurotransmitter you've got in the synaptic cleft, the higher the chance of the threshold being reached and an action potential being fired. Some drugs will inhibit the neurotransmitter, so it means that less neurotransmitter is released, therefore obviously less likely that the neurotransmitter will bind to the receptor, less likely the sodium channels will open, less likely that the threshold will be reached and the action potential will be fired. And that's it for synapses.